Okay, so this is where we left off last time. You will remember that we were the second nutrient which we were talking about in terms of recovery was phosphorus, and in the last lecture I had told you about, for example, the phosphorus recycle, that what is the source of phosphate, whether it is from the inorganic world or from the organic world. And in this slide, I had also discussed the various chemical methods by means of which phosphorus can be recovered from the waste material. And if there are no questions on this slide, we will move to the next one. And this is about the biological phosphorus removal. So apart from the chemical routes, which were presented in the last slide, the question is that whether microbial activities or certain groups of microbes, do they have the ability to remove this phosphorus which is coming from the sewage? That will be one form of extracting phosphorus and not allowing it to get wasted. Okay, so this process where microbial systems are used for phosphorus removal is called as enhanced biological phosphorus removal, EBPR for short. So what is the advantage? And then we will see the chemistry of the process or the microbiology of the process. So first of all, it is a very cost effective, environmentally friendly technology which allows you to remove phosphorus from the wastewater. And there is a very select group of microorganisms which are called as the polyphosphate accumulating organisms, which are capable of carrying out this task. So already the existing microbes which you have learned about so far are not the ones which are capable of removing phosphorus. So these special groups of microbes are the only ones which can carry out that task. Okay, and the process of biological phosphorus removal occurs, or it can occur, in two stages, and these are first anaerobic and then under aerobic condition. Okay, so the reactor in which you are going to accumulate, for example, the effluent, which is your the domestic sewage which you have collected, should first be presented with anaerobic conditions. This should be followed by aerobic conditions. What happens under anaerobic conditions is that the volatile fatty acids, which are generally generated, due to anaerobic activity, because there is not much oxygen, like the kind of conditions which we talked about when we were discussing the methane production, is that there are anaerobic digesters where these fermentative bacteria are capable of converting the carbon sources into short chain volatile fatty acids. Okay, so we are assuming that in the first stage, when you talk about anaerobic conditions, there is generation of volatile fatty acids which has occurred, which is now taken up anaerobically, and it is stored as polyhydroxyalkanate through the release of phosphorus and degradation of glycogen. Okay, so although I have listed down these things, what is happening is that you are taking up, for example, volatile fatty acids, accumulating pHa, releasing phosphorus, and you are degrading glycogen. All of these four reactions are occurring during anaerobic conditions. Then when you switch to aerobic conditions, then there is a higher amount of phosphorus which is taken up by the cells, which is from the outside. Okay, when an electron acceptor is supplied, because under aerobic condition, what you are doing basically is that you are allowing either oxygen or some other electron acceptor to come into contact with these phosphate accumulating organisms. So under those conditions, there is PHA oxidation. External phosphorus in the environment in the form of phosphate is then taken up into the cell biomass, and that is why we say that phosphorus is getting removed, because it is in the second stage that this excess of phosphorus which is lying in the waste waters is going to be accumulated in the biomass of the cell. So that is what is happening. First of all, that phosphorus is taken up inside the biomass through PHA oxidation, and there is also, it is accompanied by biomass growth, regeneration of glycogen. Okay, in the first stage there was degradation of glycogen. In this stage there is again accumulation of glycogen. Now instead of oxygen, there are other electron acceptors such as nitrates or nitrites, which can also be used as electron acceptors, and these are called as the anoxic conditions. And instead of oxygen, there can be other electron acceptors which can be introduced, except that that stage is now called as anoxic stage, not aerobic. Okay, so just let me I just want to take a call here if you don't mind. So are the students still there?
Uh, like yes, ma'am.
So
There are
just
10 students.
yeah, so just read this slide because I just have to make an important call. So just read the slide. Okay.
Okay, ma'am.
Okay. Okay. So have you read all the points which are listed on this slide?
Yes, ma'am. Welcome.
Okay. Now, in the next, we just talked about during the biological phosphorus removal, two phases. The first one we called as the anaerobic phase. The second one we called as the aerobic phase. Now, both of these are explained here in this particular diagram. So during anaerobic metabolism, as you see here, that volatile fatty acids are taken up and PHA, for example, is going to be formed. Okay. In this first phase, the phosphorus, the polyphosphates, which is a very, very characteristic feature of this group of microorganisms, they also store polyphosphates in the cytoplasm. These are broken down, and during the initial phase, the phosphorus is released into the environment. Okay, and the polyphosphate degradation also, is responsible for ATP generation. This ATP is used to synthesize PHA. So that is the anaerobic leg. Now, during the aerobic leg, which we called as the second leg, okay, what you do is that the phosphate from the environment is taken up by the cell. It is built into the cell biomass, okay, and at the same time, the PHA is broken down and glycogen is accumulated. Okay, so just for the ease of understanding, the major steps are listed down here. In the anaerobic tank, the polyphosphate is hydrolyzed like you see in this picture. It releases the phosphorus and energy, and then the volatile fatty acids which are taken up in the presence of this energy are converted into PHA, which are polyhydroxy alkanates. And I will tell you what these are. These are basically storage materials inside the cell. In the aerobic part now, there is consumption of PHA, which you have generated here as an energy source, okay, and excess of phosphate is taken up. Energy is, energy which is now available from the PHA is used to synthesize poly P or polyphosphates. So this is kind of a reversal of the steps which you saw here. Okay, and so when you say that you have biological phosphate which is being removed, it is occurring only during the second phase. Okay, during the aerobic leg. So all the terminologies are explained here. You can just read it. So therefore, the wastewater treatment plants which operate for enhanced biological phosphorus removal, they have first an anaerobic tank where there is no nitrate or oxygen present prior to other tanks to give phosphate accumulating organisms a preferential access to the simple carbon compounds in the wastewater. Okay, so overall in your mind, you just need to understand that that these sequential anaerobic part and the aerobic part is responsible for removing the phosphorus which is present in the wastewater, accumulating it in the form of the cell biomass. So you have collected, in other words, you have used microorganisms for extracting the phosphorus from the wastewater and building it into the cell biomass. Okay, then the cell biomass can be collected at this stage, and it can be broken down, and the phosphorus which is accumulated It can be treated with various salts, etc., to recover the phosphorus. That will be the part 2 of the final recovery. In the first part, there is accumulation of the phosphorus as the cell material. So I hope this part is clear to everybody. So
Yes, ma'am.
should we move forward? Okay. So now, very quickly, we look at the chemistry. So the overall process you looked at. Now we look at what is the chemistry in the anaerobic part. So what you will see here, for example, that there is oxygen is completely missing, and volatile fatty acid, acetate is given as an example to you here. So acetate is taken up inside the cell. Okay, now in the absence of oxygen, this acetate cannot be metabolized completely to carbon dioxide and water. And number two, there is also a deficit of ATP. Because remember that any carbon source, only if it is oxidized completely to carbon dioxide and water, results in the generation of large amount of energy. But under anaerobic condition, the amount of ATP which is formed is very much less. Okay, so the cells, what do? They start to break down these polyphosphates, which they are, they are only polyphosphate accumulating organisms. They start to break it down. All right, so you will see now that this is lessened by one inorganic phosphate, which is used to couple ADP to ATP biosynthesis. And this ATP in the presence of acetate will make acetyl coenzyme A, and then PHA is going to be synthesized from there. And at the same time, this inorganic phosphate, which is released during the hydrolysis of ATP, is what is released to the outside. Okay, so in understand, I think you need now in the second part, when you look at the aerobic metabolism, during this, the oxygen is available. Okay, as a result of which the excess of PHA which you have accumulated during this particular stage, okay, the PHA is converted to glycogen, which is then broken down aerobically. The TCA cycle is going to operate, and at the same time, there is a lot of ATP which is available for the cells. Okay, and this ATP is synthesized, for example, by coupling ADP to phosphate, which is taken up from outside. Excess of ATP is synthesized now. Why? Because under aerobic conditions, you make, when you break down your carbon substrate, you make a lot more ATP. This excess of ATP is now stored in the form of these polyphosphates, which you see on this side. Okay, so poly P, additional ATP is added to it, and then that is how it becomes, the molecule becomes something like this. Okay, so this is what you need to look at in order to understand the chemistry. So is this slide clear to everybody?
Yes, ma'am.
Okay, anybody else in the class, you have any questions, or is it really self-explanatory? But just pay attention to it, especially in terms of availability of ATP under anaerobic or under aerobic metabolism. So now in the previous slide, you also saw that acetyl coenzyme A, for example, let me point out here, acetyl coenzyme A is converted into polyhydroxyalkanate. Okay, what is this particular pathway? It is explained now in this slide here. Okay, so first of all, polyhydroxyalkanates are energy reservoirs in which extra carbon can be stored inside the cell. Just like glycogen can be stored, similarly polyhydroxyalkanate, which is a very general kind of a term, the basic structure is something like this. Now they can be of several types. Okay, if the R group happens to be a methyl group, then the resulting molecule is called as it will be polyhydroxy. There is a specific name to it, not alkanate, but I can't recall it right now. But if the R group happens to be this, then the resulting molecule is going to be PHV, polyhydroxyvalerates. Okay, or if this R group, what you see here, for example, happens to be this particular group, then the resulting molecule is HEX. Okay, this can be just a methyl. This is an acetate group. So it depends on what kind of an R group do you have here. Okay, so polyhydroxyalkanate, in other words, is a general term. Yeah, this is acetate, maybe polyhydroxyacetate. So, so what you are looking at on this side is how acetyl coenzyme A is converted now into polyhydroxybutyrate. Okay, so there are three enzymatic steps. I don't expect you to know the chemistry of it, but as you can see here, that acetyl coenzyme A, three enzymes will participate here: beta ketothiolase, acetoacetyl reductase, and PHP synthase. Okay, and as a result of which you have PHP. So PHP, polyhydroxybutyrate, is used as a storage molecule inside the cell. So this will allow you to understand how, for example, the acetyl coenzyme A, which you have available now, can be converted into PHA. Now the next thing, we just want to look at how polyphosphates are made inside the cell, and this slide will explain to you. So these molecules are called as polyphosphates. As the name suggests, you have multiple phosphate groups which are linked together by this this an ether linkage between phosphate. It's an ether linkage right here. So several phosphates are connected to each other by way of an ether linkage, and the resulting molecule is called as polyphosphates. Okay, you also saw that the polyphosphates, number one, they can serve as an energy source, and they can also be used for synthesis of ATP, which is explained in this particular figure, that the polyphosphate can be hydrolyzed by enzymes like phosphotransferases. So you had N, and now a phosphate is removed, so this becomes N minus 1, and that phosphate is used to couple AMP, which is adenosine monophosphate, gets converted into adenosine diphosphate. Okay, so the phosphate which you have removed from here is added to adenosine monophosphate to give rise to adenosine diphosphate. The second enzyme, which is called as ADK, or adenylate kinase, as is shown in the slide, can be used now to couple. So ADP will donate the phosphate to adenosine diphosphate to make to give rise to ATP or to adenosine triphosphate. Okay, so now in this picture you are able to understand that with the participation of two specific enzymes, the phosphates which are stored here can be used to drive the biosynthesis of ATP, which can then serve to convert a reactant into a product. Okay, so these two pathways are extremely important to know. That is synthesis of acetyl coenzyme A into a polyhydroxyalkanate and polyphosphate, which are very characteristic features of these particular groups of microorganisms, which are called as the phosphate accumulating organisms. And their role is extremely crucial in biological phosphorus removal. So now here is a very simplified diagram of an, so if you apply, for example, an enhanced biological phosphorus removal system, okay, without nitrification. Okay, so can you tell me why I have chosen the term nitrification here? Without nitrification, can anybody take a very intelligent guess on this? So any of you in the class, can you take a guess? I think there are just about 12 people today. Can you take a guess on why the term denitrification was used here? Can you speak a little bit louder, please? I can't hear you. So can anybody take a guess as to why
Ma'am,
they... Yeah, go on.
what what I feel is that the nitrification and denitrification are coupled reaction and used in the wastewater treatment plant. So in that as well, N2 is forming. So we has used denitrification term.
Yeah, and let me take you very quickly back that in this slide, please read this part very, very carefully. In the aerobic leg here, okay, if you use molecular oxygen, okay, then of course, you can carry out the reactions as I explained. But instead of nitrogen, what if nitrate is used as an electron acceptor? In which case, in the wastewater stream, if you are removing nitrate, you are also carrying out simultaneously denitrification, because that nitrate, if it is used as an electron acceptor, okay, nitrate is now going to be reduced to molecular nitrogen, and that is nothing but nitrogen, that is nothing but a denitrification event. Are you able to understand?
Yes, ma'am. I can able to understand this part.
Okay, so please pay attention, because this is really, I think, the golden, you can say, the advantage of this particular process, that during phosphorus removal even the denitrification activity can be carried out, because under, we can't say aerobic, but we can use a term like anoxic. Anoxic means only lack of oxygen. It doesn't mean lack of an electron acceptor. If you put nitrate, like it says here, alternatively, either a nitrate or a nitrite can also be used as an electron acceptor, which are called as the anoxic conditions instead of oxygen, which is advantageous because now both nitrogen and phosphorus can be simultaneously removed in the same process. That process is nothing but denitrification. Okay, so
Yes, ma'am.
yeah, try to understand, because that's the golden key here. So when you say without nitrification, that means that in this process, we are looking at an enhanced biological phosphorus removal system, except that we are going to be dealing with an anaerobic part and an aerobic part. Oxygen is there, so nitrification does not occur, and that's why we say without nitrification. So let us see, for example, if you want to integrate such a thing, phosphorus removal, you need to integrate into the existing wastewater plant. Then what needs to be done? So here is a picture which will remind you that, first your wastewater stream is taken, it's passed through a primary clarifier. There is a primary sludge which is removed, and then this supernatant is now collected, and now instead of passing it into the activated sludge part, you pass it through the anaerobic zone first. Okay, we will see as to what happens during this anaerobic zone. After spending some time, it is then passed into the aerobic zone, which is nothing but your activated sludge plant. Then it is passed to a secondary clarifier. Then you have a sludge. The activated sludge can be now returned back, and the effluent is discharged here. Okay. So now let's see what are the events which are going to occur if we make an addition of an anaerobic zone here. Okay, so what we are monitoring on the y-axis is the relative concentration, and this is the travel time through the aeration basin. So in the solid, you have polyhydroxyalkanate. Okay, as you will remember that in the anaerobic part, you have accumulation of polyhydroxyalkanates. Okay, so with respect to time, as more and more time is spent in the anaerobic zone, the accumulation of polyhydroxyalkanate is going to go up. And that's what this figure means. Okay, and you see here the broken lines here are nothing but these are glycogen, because remember that glycogen is broken down during this phase, PHA is accumulated, and the small dots which you see here, for example, are the volatile fatty acids, because during this process you will recall that during the anaerobic process the volatile fatty acids are consumed, and that's why their level is going low in the anaerobic zone. Okay, and this is your phosphate, and obviously phosphates are going to be accumulating again, as I said, that you are eventually, because polyphosphates are broken down, so in the initial phases the phosphorus is now added, or it is released to the outside of the medium. That is what these four lines represent here as a function of time in the anaerobic zone. Is this clear to everybody?
Yes, ma'am. It is clear.
All right, and now let's see what happens in the aerobic zone. Okay, so now after giving a certain residence time in the anaerobic zone, this is what happens to these four parameters. Then under aerobic conditions, what you are looking at, for example, is number one, because polyhydroxyalkanates are going to be completely consumed, glycogen is going to be building up, and then volatile fatty acids are anyway, they have gone down, so nothing happens to them. But at the same time, when you have when you have phosphates which are released, okay, these phosphates are now released right here. No, sorry, phosphate is this particular term. Okay, which are the smaller terms here. So if we look at the phosphorus released to the outside, all right, so it goes down, because now the concentration goes down in the extracellular medium because they are taken up by the cells. So I hope this is clear to everybody. If I have made a mistake, maybe some of the things I may not have said correctly earlier, please take care. And so we see glycogen is restored. Polyhydroxy alkanates are broken down during this stage. That is why glycogen is accumulated, and phosphorus, which is present now, also goes low because it is taken up by the cells and it is accumulated as cell biomass. All right, so that is what therefore is expected to be seen if you were to incorporate the biological phosphate removal. I hope it is clear to everybody. So should we move to the next slide?
Yes, ma'am.
So in this, all right, I mean, it is what I said just now. Okay, so please read it. It is only to summarize these points, and it's exactly what I have said just now. You only need to read it. And now here is, here is a phosphorus balance which has been given for a typical municipal wastewater treatment plant in Germany, where they have incorporated the biological phosphorus removal. Okay, so what you see is that in the influent, this is your wastewater stream, which is now influent into the plant. So if you look at, for example, how much of phosphorus is present in here, it is approximately 1.8 gram, which is capita per day. Okay, so that is the total per day in terms of how many people are resident in that particular community per day. So the influent receives approximately 1.8 grams of phosphorus, which is now at time zero, it is considered to be 100%. So please try to understand what that term means. Okay, so total amount of phosphorus which is added, per person per day is 1.8 gram. Now in the primary sludge, PS refers to primary sludge. So when you carry out the primary settling and you remove the primary sludge, the amount of phosphorus which you are losing here is 0.2 gram, which is actually 11% of the total 1.8 gram. In the secondary sludge, okay, which you remove after the activated this thing, you remove approximately 28%, which is 0.5 gram. Okay, however, if you now, for example, through enhanced biological phosphorus removal, 0.9, that is approximately 50% of the phosphorus which is coming in here can be removed if enhanced biological phosphate removal followed by precipitation is incorporated into the wastewater stream, so that in the secondary sludge you are only going to lose about 28%. Okay, so in other words, all right, by incorporating an this is enhanced biological phosphate removal, by incorporating an EBPR in the existing wastewater plant, nearly 50% of the incoming phosphorus is captured now. So it is not lost. Only this much is going to be lost, which is in the form of primary or a secondary sludge. Anyway, the secondary sludge, if it is taken up, the sludge is thickened, and if it is applied into the field as a biofertilizer, then also quite a lot of it is recovered. Okay, and finally in the effluent, you see only 11% of the incoming is lost. Rest can be recovered, and it can be put to various uses, and from the cells also phosphorus can be removed. Okay, so is this clear to everybody as to what we are trying to say here?
Yes, ma'am.
All right, so make sure you also understand, because these are not events which are occurring in isolation. Eventually, when you learned about the microbiology of these bugs, when you learn about the biochemistry of these bugs, you want to incorporate them into the existing wastewater treatment plants. Okay, in order to address the problem of energy recovery or nutrient recovery, such as nitrogen and phosphorus. Now, here is something which is from a paper, and I only want you to appreciate that in order to remove the phosphorus along with the nitrogen, okay, because in these processes, or in the slide which I showed you last time, we are recovering phosphorus, but we are not recovering nitrogen. But what if you want to plan to recover nitrogen along with the phosphorus? That can be done by supplying not molecular oxygen, but nitrate can be supplied as an external electron source. Okay, then you can carry out recovery of nitrogen and phosphorus at the same time. In order to do that, in this paper, there are two different schemes which have been applied. Okay, so in the first scheme, it is called as A2O process. All right, now A2O means that A stands for anaerobic, the second A stands for anoxic condition, O stands for aerobic condition. Okay, so please try to understand, because in a reactor, for example, these are all lab scale experiments. So what they have done is that the wastewater is collected and it is subjected to an A2 process, A2O process. So once it is subjected to anaerobic condition, all these bacteria, fermentative bacteria, will release the small chain short volatile fatty acids are going to be released. Then when you pass and under anaerobic condition, also you have phosphate accumulating organisms which are going to accumulate now. Okay, and they are able to take a phosphate, they build a polyhydroxy alkanate. In the second stage, you have anoxic condition, that is, you are going to supply, it's not aerobic condition. Anoxic means lack of oxygen but presence of nitrate as an electron acceptor. So once you give nitrates in this, you are carrying out actually the denitrification. Those nitrates are going to be converted now into molecular nitrogen, and phosphorus is also simultaneously removed from the effluent. Okay, this effluent then is passed into the aerobic part, where you have the activated sludge which is occurring, and eventually the wastes have been subjected, and then you go to secondary clarifier, you collect the waste activated sludge, recycle a part of it, and this is effluent. So that is how you understand the sequence now here. What is the purpose of anaerobic reactor? What is the purpose of anoxic? What is the purpose of aerobic here? And this is why both nitrogen as well as phosphorus is removed, because you are supplying here, in the wastewaters, the nitrates which are already there are going to be used as electron acceptors by the phosphate accumulating organisms. So is this clear to everybody?
Yes, ma'am.
All right, and the next stage we say reverse A2O. Okay, by reverse A2O, what we mean is that first we have put anoxic here, then we have put anaerobic here. Aerobic stays as it is, but reverse A2 means first you carry out anoxic, then you carry out anaerobic. So under anoxic conditions, okay, under anoxic condition, you have different sequences. You should be able to interpret them. That under anoxic condition, you are going to remove the phosphate, etc. The nitrates are going to be removed first by these, and then under anaerobic condition, you have the phosphate accumulating microorganisms which are going to be occurring, and then aerobic condition, and then quite a bit of it is now recirculated. So whatever could not be completed in the anaerobic part of the leg with respect to phosphorus removal can be carried out in this particular part, because remember that this liquor is now going to be recirculated in the influent, unlike here. Okay, so make sure that you pay attention to this. So this mix, whatever comes out of here, goes back into the anoxic tank, so that the remaining nitrogen is also removed in the influent, which you did not get a chance to do earlier. Okay, so therefore pay attention to how both the phosphorus removal and nitrogen removal can be integrated into the same process. Okay, so with this, we complete our discussion on the nitrification, denitrification, anamox, phosphorus recovery, simultaneous recovery of nitrogen as well as phosphorus. So with this, I hope it is clear to everybody, and try to read, the reading material is already circulated. So I'm going to move to the last lecture, which is now called as risk assessment. So is this clear to everybody now? Kaushik, are you here? Amritanshu, Kaushik, you're logged in. Are you here? Are you listening to the lectures? Gopika, Have you people returned to campus?
Yes, ma'am. I think um, I have returned, ma'am.
And what about Kathy? Have you returned to the campus?
Yes, ma'am.
Okay, so where are you taking the classes from? From the hostel room?
Yes, ma'am. Hostel room.
Okay. Okay. So is Kathy, is it clear whatever I discussed in the lecture?
Yes, ma'am. It's clear.
Okay. And please go through now. The exams are just round the corner, and read the reading material. If you want me to, a couple of times more, we can sit together apart from our presentation lectures, and we can have doubt sessions. We can have some extra hour with you, so that you can come back and you can have, I can take any questions from you. So now let me come to risk assessment, which is our last topic. Okay. And what I'm going to do is that I'm also going to leave, there is an audio file which is attached here. Of course, you will listen to the lecture, but you can. Okay. So now, what is risk assessment? All right. Now, risk assessment is actually whenever a chemical or a hazardous infectious particle like a virus or a bacterium is released into the environment, then you would like to know what is the probability of somebody catching an infection, or what is the probability of somebody falling ill as a consequence of exposure to that particular chemical or a pollutant. So to put a quantitative measure to that term, all right, is called as the risk assessment. Okay, so is this clear to everybody? So let me put down all of these points in order to make you understand. So whenever we do, environmental biotechnology, a lot of data is gathered on the occurrence of either pathogens or pollutants or toxins in the environment. Okay, you say, well, so many milligram per liter lead is present, or virus particles, or 100 virus particles per liter of water is present in the pond water, or whatever. So a lot of data is gathered. However, one has to interpret this data, and this will be critical in making a decision about the potential health risk and a corrective action to be taken. Okay, so quantitative risk assessment is an approach which allows a quantitative expression of risk in terms of the probability of infection, illness, or the mortality from the pathogen. Okay, and number two, the data which you have, it allows the decision makers to determine the magnitude of the risk and weigh out the cost and benefits of the corrective action. Okay, so overall, I expect that the students will try to understand, because then the following slides will become very, very clear to you. So is that clear as to what is being said? Very quickly, yes or no? So I don't see any response from the students. Are you there? So Kathy, are you still there?
Yes, ma'am. I lost my network.
Himanshu, what about you? Himanshu, are you there? Ratnesh? So should we continue the lecture or not? Should we continue the lecture or not? Can you hear me?
Yes, ma'am. You can continue.
Okay, so risk assessment, is always a normal part of many, many activities. And I'll give you some common examples to let you know where all risk assessment is usually carried out. Number one, on the Wall Street, where you have people playing with stocks, they make an assessment of the financial risk. If you were to invest so much, then what is the likelihood of a stock price to be going up to a certain level? Okay, based on so many other parameters which are taken into the account. But then what you are doing there, the Wall Street analysts are also making a risk assessment, a quantitative risk assessment. Similarly, the insurance companies are all the time making a risk assessment. If somebody is very old, like over 50 or something, then if you were to purchase a policy, for example, for 1 crore or something, then the policy premium is calculated on the basis of what is the risk that a 50-year-old has on falling sick or having cancer and things like that. So those insurance policy premiums are going to be calculated based on the risk factors. Okay, so that is another place where the risk is always calculated. Similarly, the regulatory agencies, okay, so they estimate the risk of deaths from the nuclear plant accidents. Similarly, industrial emissions, they estimate the incidence of cancer, for example, that if I'm releasing 20 parts per billion into the environment, and based on my scientific data, that is, which is called as a dose response curve, what is the probability that if people who are living in this neighborhood and they are exposed over a certain period of time to 20 ppb of lead, what is the probability of their contracting a cancer? So they do a risk assessment. Okay, so in a similar manner, in environmental biotechnology, also risk assessment is carried out. Okay, so is this fundamentally clear to everybody? So we can say that risk assessment, therefore, is a process of estimating first the probability that an event is going to occur, and number two, the magnitude of its adverse effect. Okay, how much? What is the probability of somebody catching an infection out of the presence of, let's say, 2 viral particles in 1 liter of water? Okay, so it also gives you a probability or the magnitude of the effect. Is it 20%? Is it 40%? Is it 60%? Over a specified period of time. Okay, so I hope that this is now clear as to how risk assessment and let me just come back before we discuss this table. So when you carry out overall risk analysis, there are 3 parts to it. The first is called as the risk assessment part, which is science based. Then the second part is called as the risk management part, which is policy based. However, the management of that risk is based on the scientific data which you have collected. Okay, and both risk assessment and risk management are then communicated. So risk communication to non-experts is the third part, because these could be the policymakers, or you want to interact with the general public and pass this information of risk to the general or to regulatory bodies, whereby the standards are going to be set. So whatever you do in the lab, whatever policies you develop, you finally communicate risk to the public. That is called as the risk communication. So overall, the risk analysis, therefore, has 3 different components. Now, let's look at the first part, and then I will just very briefly explain all of these. So when you look at the first part, which is called as the risk assessment, we say it is science-based. It is based on the facts or the actual data which you have collected from the field. All right. And risk assessment itself has 4 components. The first one is hazard identification. Okay. Now, before you talk about, how much you are at risk by exposing yourself to lead or to viruses or to certain microorganisms, you need to identify that particular hazard. And that is called as the hazard identification. It could be either a contaminant, it could be a metal, a heavy metal, it could be a solvent, which is a hazard, it could be a pathogen, and documenting its effect on humans, that yes, this particular virus is an agent of harm to humans. The second is exposure assessment. That is, once you have identified that, okay, exposure assessment is what is the concentration to which you are exposed to. So lead could be in milligram per liter in the water body. Okay, but then if you are drinking 2 liters of water in a day, then what is your exposure going to be? That is what we mean here. That exposure assessment is the concentration of that in the environment and how much you are going to take it up. For example, concentration of salmonella in meat and the average dose a person could ingest depending on how much meat they are going to eat. They will be exposed to a certain level of salmonella. Somebody who is purely, purely a meat eater, doesn't take any vegetables, is likely to be exposed to a greater level. So exposure assessment is that part. So is that clear as to what we mean by exposure assessment?
Yes, ma'am.
Third is the dose response curve. Okay, this you already know, that if somebody is exposed to 4 parts per billion or 10 parts per billion or 15 parts per billion, what is the dose? So dose response curve you need to develop for that particular hazard which you have identified, how much exposure you already know. Then you need to develop a dose response curve, and finally what is called as a risk characterization, which is estimating the impact, illness or the death of a microbe, or because of a microbe or a chemical, based on the severity of the effect and the amount of exposure. So in other words, by risk characterization, what we mean, what is the probability of a person developing an infection after having been exposed to a certain exposure level? That part is called as the risk characterization. Okay, and all of these one has to do under the risk assessment. The second is the risk management. Now, this is something which is only policy based, and here the regulatory options are evaluated, which includes the social, political, economic issues, the engineering problems inherent in the solution. Okay, so let us say the risk, etc., is you already know that if you are exposed to lead, for example, 100 ppb, from the dose response curve, you already know that the probability of somebody getting exposed, with the 20 ppb, for example, the response generally is so much, and finally you predict the risk. This is something which can then be communicated to an engineer, this data, and then they are going to find a solution. So let us say you are saying that in a water bottle, after I do my dose response curve risk characterization, that if you leave 4 E. coli particles in a 1 liter bottle, okay, then the risk is so much. There is 50% risk of contracting an infection. But if you leave, for example, only 2 E. coli particles in 1 liter of water, then the risk is 20%. Okay, then these regulatory options are going to be evaluated by economic issues, by social issues, etc. Then an engineer is going to find a solution, that do I want to take a 20% risk of not setting up a strategy whereby I'm going to clean up the water to a level where only 2 E. coli particles are left. Okay, because 4 in his opinion is too much, because then there is a 50% probability of somebody getting that particular risk. But if there are only 2 E. coli, then there is a 20% probability. So engineering problems and solutions can be devised, therefore, on the basis of the risk which have been characterized. Okay, so problems, solutions, etc. to the problems can only be devised once you have a science-based data in front of you. And then finally, the third leg, which is the risk communication. So eventually, based on the risk, this information is communicated from experts to the non-expert audience. And on the basis of the science, on the basis of the various management, and on the basis of the risk communication, there are regulatory agencies which have been created, such as FDA, which is Food and Drug Administration, EPA, Environmental Protection Agency, Occupational Safety and Health Administration, etc. So the function of these agencies is to use this risk data to pass federal and state laws for protection of public health as well as environment. Okay, so is this part clear to everybody? Because this you can consider as the key slide in the lecture. So is this part, is the information on this slide clear to everybody?
Yes, ma'am, it's clear.
All right. So now, although very briefly I told you what the regulatory agencies, based on the science, based on the management, and based on the risk assessment, the role of these regulatory agencies is listed down here. They will set a standard for the concentration of toxic chemical or microorganisms in water and food, that this is something which is acceptable and this is something which is not acceptable. Okay, and They also assess the risk from the release of genetically engineered organisms, because today, a lot of recombinant DNA work is being carried out, and that poses a threat to the environment. So they will also tell you what is the risk if you use an engineered tomato, for example, or you are releasing, for example, your BT cotton, which is a cotton crop which already has an insecticide which is made by a certain bacterial gene inside. So if you release this into the general population, this particular genetically engineered crop, does it pose any threat? Okay, so those risks are also calculated and they are conveyed to the general public. They conduct a baseline analysis of the contaminated sites to determine the need for bioremediation and extent of cleanup which is required. Then they perform a cost benefit analysis of the cleanup or the treatment option. This is all the job of these regulatory bodies. And things like FDA, because you will recall in the recent days, all the vaccines which had been developed, against our this coronavirus, there was an FDA approval which was to be required, which was to be given, or ICMR, which is the Indian Council of Medical Research, had to give an approval first based on the data which they looked at, based on when the vaccine is given to so many people, how many people responded well, etc. And then they decided that, okay, it is time to release the vaccine to the community. Okay, they also perform a cost benefit analysis. They develop a cleanup goal. They construct a what if scenario, that what, if this happens, then what is it that we are going to do? So regulatory agencies are going to set up that. They evaluate new technologies for prevention, control of hazards and risk, articulate the community public health, etc. Okay, so I think in that chain, you need to very carefully understand that how all of these, the science, then the management, which itself relies heavily on the science which you have generated, and how eventually it is going to be communicated to the regulatory agencies, and then how these standards are put into use, how they are followed by different countries, different, global standards are set. It's a combination of all of these three factors. Okay. So now, very quickly, I will give you some details which are involved in each one of these. Like in the risk assessment, I talked about hazard identification. Okay, so now, we talked about in hazard the quantity, the exposure, or whatever. But there is one point which you need to understand, that in this figure which you see down below, for example, are the kind of studies which you need to carry out in order to understand the hazard. What is the kind of the assessment of the hazard, what kind of studies need to be carried out. Okay, now on the y-axis, if the risk is about 1 in 10, okay, then clinical observations need to be carried out. Okay, if you don't understand, please interrupt me. So if the frequency of occurrence of that particular disease is very high, or the percentage is very high, okay, then you can do it under clinical settings. That is, you have like 100 patients which are in the bed, like through coronavirus, etc. Because the percentage of occurrence of infection is much more. It is 1 in 10. Okay, which is like 10%. Then clinical, it can be done under clinical setting. If the percentage reduces, okay, if it is only 1%, then you cannot carry out the assessment under clinical settings, but you need to do on an epidemiological study. That is, a large population is going to be surveyed now in order to find out how many people are infected. Okay, if this particular risk is only 1 in 100, then epidemiological studies need to be carried out. Okay, now if the risk further reduces to 1 in 1,000 or 1 in 10,000, then very large epidemiological studies need to be carried out. That is, entire populations or villages need to be taken, and then you need to find out what is the exposure, what is the assessment, etc. And finally, if the probability is very, very low, then you need to carry out studies from animal, because it's very, very difficult to carry out such studies, on an epidemiological basis. Then you are going to carry out, you may give out very large doses of that chemical or to viruses to animals, okay, And then you may even extrapolate. So if, for example, if the animals are exposed to 100 ppb of lead, 100 animals are exposed to 100 ppb of lead, okay, then if they were exposed to only 50 ppb, then what is their response going to be? So you are going to make an extrapolation from this kind of an animal study. So this is a very important component, because what it tells you is that as, for example, you move down, so this is kind of if the risk of infection is in percentage or parts per million, parts per billion, or parts per trillion, as it tends to go down, okay, the kind of studies which you need to conduct are also going to be very, very different, and one needs to be aware of this. So the next part is exposure assessment. Okay, now in exposure assessment, what needs to be understood is that what is the intensity, what is the frequency, and what is the duration. Now by intensity, it's like if somebody is exposed to radioactive intensity. Okay, that means are they exposed to, let's say, 20 microcuries, or are they exposed to 5 microcuries, or are they exposed to 0.1 microcurie? Like people who are working in the nuclear power plants. Okay, because when you talk about risk assessment, you are not only talking about chemicals, pollutants, you are also talking about viruses, you are talking about radioactive exposure, so on and so forth. So in the exposure, knowing the intensity is very important. The frequency, how often is somebody getting exposed? So somebody who only goes for a routine examination of the nuclear power plant is not getting exposed to 20 microcuries. It could be only once in 2 months, but the worker who is there is getting exposed to 20 microcuries almost twice every week. So therefore, the frequency is going to be important when you talk about exposure assessment. So some figure needs to be there in order to take care of that. And the duration, for how long. So you do, for example, twice a month you are exposed, but for how long? Are you going to be examining that for 2 hours or only for 2 minutes? Okay, so in other words, when you talk about exposure assessment, all of these factors are going to be extremely important. So is that point clear to everybody? I mean, there is no uniform for everything that you are taking, for every chemical, for every pathogen, for every radioactive exposure you are taking. All of these things need to be incorporated into the exposure assessment term. And finally, you have the dose response curve, which looks something like this. So this you have to scientifically develop for every pathogen under laboratory conditions. Okay, so if you are exposing, your exposure you already know, but a response curve has to be generated. So if you are exposing somebody to 5 milligram, to 10, to 15 milligram per kilogram of body weight, etc., what is the response going to be? Okay, so this this response curve is going to be used in order to finally calculate what is the risk factor that a person has. Okay, so I hope all of these terms are clear to everybody, that what do we mean by hazard identification and at what scale you should be operating depending on what is the risk, at what level is the risk. Exposure assessment, then what all you need to know. In the dose response curve, what all you need to know. Do you need to extrapolate it to low level exposures also? Because at very low level maybe you can't see directly any kind of an effect, so you will expose coffee or saccharine. Rats were exposed to very high levels of these, and then the data was extrapolated to the low level exposures. Okay, so all of these things need to be looked at. And here is something which is, if you don't get any response, for instance, then what can be done is that, you can work background. Now, for example, if you are eating lettuce as a part of the salad, you could be getting exposed to certain bacteria like salmonella, but then the concentration of salmonella in 1 gram of lettuce is so low, okay, that it is very difficult to estimate that number. So what you can do, you can go to the original source where the lettuce was harvested, in that soil, how much was the exposure, etc. And from there you can extrapolate then to what is your risk, for instance. So is that point clear to everybody?
Yes, ma'am.
So that is what what is an example of estimating an exposure, and this is done from what are called as event trees. So as I said, finally, what you are trying to look at is the salmonella, which is a bacterium which can be taken in when people are using unwashed salads. If they are eating, then there is a possibility of a bacterial infection. So salmonella, which is ingested per person per year, okay, will lead to, for example, exposure to 2 times 10 to the minus 4 colony forming units per gram of lettuce. Okay, so where does this number really come from? Obviously, it cannot be directly computed. By colony forming unit, what we mean is that it refers to one bacterium. Okay, why a colony? Because if you take that sample and you plate it out on a petri dish, then wherever one bacterium had attached itself to the petri dish, there is a colony which is going to be observed. After so many doublings overnight, the bacterium will undergo so many doublings, and you will also see that there is a colony. So every colony is actually indicative of one particular bacterium. Okay, obviously such a low number cannot be estimated. So you are saying that a person who consumes, who is eating lettuce, their infection is going to be, exposure is 2 times 10 to the minus 4 colony forming unit per gram. Obviously, it's difficult to estimate. How did I get this number? Then I work on what is called as an event tree. I work backwards. So we say that, okay, when you start out with the raw sewage contains 2.89 times 10 to the 7 salmonella per ton of raw sewage. Okay, and now the sludge which is now formed from the raw sludge, okay, because when you remove in the primary the effluent etc is removed, very little bacteria goes away. Almost 83% is carried over. So all these bacteria which were now in the sewage are passed to the sludge. 83% of the bacteria, they remain in the sludge. Okay, so this is 83% of this number, which is 2.4 times 10 to the 7 colony forming unit per ton of this sludge. Now, through anaerobic digestion, you carry out anaerobic digestion, etc. And then the sludge, which is now left behind, okay, carries 1% of this, which is actually 2.4 times 10 to the 5 colony forming unit. The rest is left behind. Okay, the rest is completely gone away. So in that solid part, you have 2.4 times 10 to the 5 colony forming unit per 10 of the sludge which is left behind after anaerobic digestion has occurred, or in the solids part. Okay, so let's say this is taken and it is applied in the field. Now it is directly gone to the field, so there is some kind of a dilution. After incorporation into the soil, it's about 104 dilution. So per ton of soil, we have 2.4 times 10 to the 3 colony forming unit. Okay, in the soil, it undergoes some kind of a decay, and then you have only so much of left after 5 months, colony forming unit. And now if lettuce is grown in this particular soil, so much is taken up. So you get, for example, 4.8 times 10 to the minus 4 CFU per ton of lettuce which is produced. And if a person eats so many grams, is consumed in a year, then per person, the number works out. So in other words, in the case that the event is very, very small, one can go back to this kind of a calculation and then figure out, for example, what is the extent of your exposure going to be. So these are called as event trees. Okay, so now here is, for example, finally, what do I do with all the model? So so exposure level, etc. you have determined. The third part is the dose response curve. Okay, so in this dose response curve, what you will see is that this is the dose on the x-axis, milligram of that contaminant to which somebody is exposed to per kilogram body weight. So this could be 5 milligram, 10 milligram, 15, 20, 25, and 30. And this is the response. Okay, so the percentage of so 100 people were taken and they were exposed to so many milligram, so many people. So 20% of them developed if they were exposed to so many milligram of this particular. So such a dose response curve is generated now for that particular contaminant. Okay, so when you examine this, it follows a Poisson distribution here. Okay, so just like your normal distribution for something or your exponential curve, so the dose response curve for this particular case study follows a typical Poisson distribution with its constants, which are defined as alpha and beta. Okay, so now based on this particular data which you have, you can calculate what is the probability of infection from a single exposure, which is called as P, which can be calculated based on the dose response curve and the constants in it. So N is the number of organisms which are ingested per exposure. Alpha and beta are the constants which are derived from this dose exposure curve. Okay, so is this point clear to everybody?
Yes,
Yes,
ma'am.
ma'am.
Okay, so from the dose response curve, in other words, you need to know what kind of a distribution it is following. Is it following, because there are only, two types which have been observed. Okay, in this table are given. For example, if a person is exposed to echovirus 2, echovirus 12, and there is a certain level of exposure, then the dose response curve has been seen to follow a beta Poisson distribution with the constants, model parameters as alpha and beta, which are derived from the distribution. Similarly, if a population is exposed to rotavirus, then the dose response curve shows a typical beta Poisson distribution. Or if it is exposed to poliovirus 1, there is exponential distribution, so on and so forth. Okay, so depending on what your dose response curve, what kind of a model fits best, you derive certain constants of the models and you can calculate what is the probability of infection. So let's take a case study in order to understand that. Okay, I will skip this slide and you can listen to it. It's pretty clear. Okay, so let me just take a pause here. Okay, we learned about the dose response curve and how to calculate. Now at this time I want to introduce another term which is called as a microbial risk assessment. Okay, because so far in the previous slides what we have looked at was, let's say, exposure to a certain contaminant, a certain pollutant, a certain chemical, etc. All right, maybe viruses also. But in the microbial risk assessment, the hazard identification, there are some other unusual events which are likely to occur. And that's why it is dealt as a special category. Okay, now for example, somebody takes a contaminated drinking water, it is ingested, okay, and there is infection. However, the person could be either asymptomatic, like when you saw the coronavirus, there were people who were carrying this infection, they were exposed, they were carrying the infection, but then there was no manifestation of it. So there is a distance, between the infection, between the exposure, the clinical symptoms, and finally the mortality. So in a microbial risk assessment, a lot of these factors also have to be figured in order to arrive at the final mortality number. Okay, or a person could be simply infected but could be asymptomatic, and they are going to carry out the secondary infection and all that. So I mean, just be aware of it, because then in the model, you need to make all the necessary changes, because somebody could have been exposed to so much, but it's not necessary that they are going to, maybe only a fraction. Supposing there are, there are 5 viruses which a person is exposed to, out of which only 2 viruses are capable of multiplying. The rest have been even just taken away by the antibody system of the body. And only these 2, there is only a fraction of those viruses which is capable of causing an infection, in which case the distribution curve will look quite different than what you would assume. Than what you would assume, for example, if a whole integer, if 5 or 6 were exposed. Okay, so in microbial risk assessment, therefore, because of all of these complications, certain other factors have to be figured in. Okay, so now, based on what I discussed, here is just an example which will allow you to understand this very well. Okay, so I'm going to read it out with you, that both hepatitis and viral gastroenteritis, they are caused by consumption of raw or cooked clams as well as oysters. Some people take seafoods, etc., and they are inadvertently exposed to viruses or hepatitis virus or gastroenteritis viruses, etc. Okay, so if a person is consuming that seafood, what is the probability of their getting infection or they are dying because of eating oysters? So based on all of this, I'm going to allow you to understand as to how the actual risk assessment can be used. Okay, so in this study, the concentration of the echovirus 12 was found to be about, so this is a typical virus, echovirus 12, which resides within the oysters. And what was found is that it contains about, on an average, there are 8 plaque forming units, which is nothing but the virus particles. So there are 8 viral particles which are per 100 gram in oysters collected from some particular site. So the question is, what is the risk of becoming infected and ill from this virus if the oysters are consumed? Assume that a person usually consumes 60 gram of oyster meat in a single serving. Okay, so I hope you understand what the question is all about, that people who are on seafood, okay, you just want to calculate what is the risk of that person falling ill, given that the abundance of this particular virus in the oysters is of a certain level. Okay, so is this point clear to everybody? Please, very quickly, I think this is also a key slide in here. So should we go forward? Is it clear to everybody?
Yes, ma'am. But how do we know which model to use?
Exactly. That is based on the dose response curve. That is the data which is given to you. Somebody has already done that work scientifically. All you are doing is that you are carrying out the risk. Dose response curve data is already available with you. So what you know is that what is the exposure level. So we are saying that if a person consumes 60 gram of oyster meat, okay, so we are saying that if there are 8 plaque forming units which are present in 100 gram, then if a person is consuming 60 gram, okay, then N exposure is only 4.8 plaque forming units. Okay, very simple. In 100 gram you have 8, so in 60 gram you have only 4.8 plaque forming units. From the dose response curve which is available to you for echo 12 virus, it follows a Poisson distribution that has been established. The value of constants alpha and beta is already known to you. So the question is, what is the probability of infection, which is P is equal to 1 plus N. Remember in the last slide I had shown you, it is 1 plus N divided by beta, and this is alpha. So you plug in the various constant. N is the exposure which you have calculated, capital N, because this is the probability function right here.
Okay.
Okay, so P is equal to 1 plus N, and you have calculated the exposure is 4.8. Beta from the model is already given to you, 186. That's why I put in this number here. Alpha, which you are supposed to plug in here, is this. So you have 9.4. Okay, so this is the probability of infection. Now, very interesting, you move on. If the percentage of infection that results in the risk, because infection doesn't mean that there is a clinical illness. So you have been infected, you have been exposed, but then you may not develop the symptoms at all. But they have calculated that there is a 50% probability. If that is the case, then the risk of clinical illness becomes 50% of this number. So the number which you get is right here, which is 50% of this number. If the case fatality rate, somebody becomes ill also, but the fatality is only 0.001%. If that is the case, okay, then the clinical illness is so much, you have the number here, 50%, but only, 0.001% of fatality is this much. So now you are able to calculate the risk of mortality. So the risk of mortality is so much. If a person consumes oysters 10 times in a year, then then the annual risk is going to be so much. Okay, so all of these numbers, in other words, now, having integrated all of this information, etc., you can take a call and you can inform a person that, look, this is the risk which you are facing. So if in a year you're going to consume 10 times in a year, you're going to eat the sea meat, the oyster meat, for example, then your risk of infection is so much, or your risk of mortality is so much. Okay, so that is what risk assessment is. It is actually to give a quantitative term to the exposures, and once you have a quantitative term, as I said, the policymakers or an engineer can take a decision, because if you have so much. I have another example here, uh, which is like, for example, in a wastewater treatment plant, if you know that if we leave so many of certain viruses in the water, then the risk of infection is so much. Then the person, the regulatory, in consultation with the regulatory bodies, he or she has to take a call as an engineer that, look, my treatment strategies must include this part also, so that there has to be an additional filtration step, so that now the number of bacteria in the drinking water comes down to only 2 from 6, because I have looked at the dose response curve. I have also seen what is the risk of somebody falling ill or dying. So I need to develop a strategy. I need to integrate it into the existing wastewater treatment or uh, any other environmental scenario, and based on that, the federal laws, the agencies are going to set up rules and regulations. Global standards are going to set up. Okay, so is that point clear? This is how important it is. Okay, so in this entire exercise, what you are looking at basically is to put a quantitative term and to allow people to make a very informed decision. So I hope it is clear. Okay, yes or no, quickly.
Yes, ma'am.
Okay, so read that. I will also that it's already in the chapter. The reading material is there. So read, just about 7-8 page long chapter, more of a common sense. You just need to put different pieces together in your mind. So in summary, risk assessment is a major tool for decision making in the regulatory domain, both regarding chemicals, microbes, viruses, radioactivity, or what have you. Okay, and the results of such analyses can be used to inform the managers of the probability and the magnitude of the environmental impact, which is resulting from exposure to different contaminants. One can also use this to weigh the cost and benefits of control options and to develop certain standards. Okay, then in that chapter, risk assessment chapter, which is in the textbook, there are a number of questions given in the question bank. So please see those also. So any questions on this? Is this clear to everybody? So these are only 15 slides, and there is a good chapter as a reading material for this. So please read this just to allow you to understand it better. So I'm going to stop here now. Any questions on this lecture?
Ma'am, in slide 12.
Slide 12. Okay. This one?
In the box of case study,
Yeah.
saying that they have calculated the annual risk, PA.
Yeah.
How do I get this formula? Because I saw that they.
Exactly. So I have not dealt it, but in the chapter it is dealt with, and it is all of these things are set in the probability of developing. But for 1 year, they have derived this formula. That is in the text. That is in the chapter. If you look at it.
Okay, ma'am. So this formula would be 1 minus P raised to.
The probability, what you calculate, for example, here.
Yes, ma'am.
Okay, the probability, and then raised to power 365. But of course, they have here 1 minus this much, raised to power 365.
Yes, ma'am. And just in the line before this, they stated that if a person consumes 10 times a year,
Yeah.
so in the formula, it is nowhere accounting for 10 times. So if a person consumes 20 times, will this formula be the same?
I think the total, then the exposure time is going to change. In my opinion, that the exposure time, let me work it out myself also to see and then I will just post it back to you.
Okay.
Because this number will certainly change, because total exposure level is going to change.
Yes.
Okay, then the number changes here. Let me work it out also. But other terms, are they clear to everybody?
Yes. Rest all was clear.
Okay, and of course, this is a case study where it follows a Poisson distribution. Then there is another case study which they have done on rotaviruses, where it follows an exponential curve, where the constant is R and the value of R is also given. And so accordingly, one can calculate the probability. Okay, so I'll share that chapter with you. It's in the same book, which is the environmental microbiology book. So any other question from anybody? And then please come back, because in the doubt session, it's a classroom examination. So I will, whenever you feel you want to have 2 classes online, for a doubt clearing session, there is no problem with me. Let me know, and we can just collectively, we can just choose a couple of hours, come back and do this.